norms. So norms is again set up by the departments, various departments. Especially if you talk about the India, then for the India, it is the pollution control board. It may be the central pollution control board or state pollution control board. So they decide what is the range. So if you see the pH contents for the drinking water, for the agriculture water, or for general use, what will be the pH? So they give the norms of it set between the 6 or between the 6.5 to the 8. It will be the drinkable. It will be used for the agriculture purpose and all. So this is the norms is set up. Okay. Now when the wastewater is generated from the industry, now we would like to know what is the concentration of it, the contents. It may have the organic contents, it may have the inorganic contents, it may have the oil types. Okay, whatever it may have the pathogens types. So depend on the what type of the industry it is, what they are manufacturing, and on the basis of that one, they are producing the waste. So whatever be the waste which is generated, that we need to know what is the content in it and what is the value of it, what is the number. So what will be the pH, what will be the BOD, what is the COD contents present in the wastewater. And on that basis, we will choose the technique or we choose the process to treatment of it. Just like this is the theoretical initially when we are designing the any plant, then it will be the theoretical one. So this is the process industry. This type of the waste is generated. This may be the sample. This may be the concentration of the waste. And now we need to reduce it. And for that one, what will be the process that we need to choose, whether we have to give the organic treatment, whether we have to give the inorganic one, whether I have to choose the aerobic process, whether I have to choose anaerobic process, should I only give the primary treatment or it is need to secondary and tertiary treatment. Okay, so all these things we are decided on the basis of what is the concentration of the waste, what type of the waste it is. So one of the tests that we are carried out, most of the industries, though it is a lengthy process, but yes, we are using that test which is called a BOD. Now what is that BOD? BOD stands for the biochemical oxygen demand. If you see on this one, okay, okay, I just yes, the BOD. What is this BOD? It is a biochemical oxygen demand. What is this? It is a biochemical oxygen demand. Most of the times when we the question asks what is BOD, we say it is a biological oxygen demand. Remember the word, it is biochemical oxygen demand. Now what is the meaning of biochemical oxygen demand? Okay, before that we will look about another test that is carried out. It is the chemical oxygen demand, COD. Also, we are carried out the total organic carbon, which is also called TOC, total organic carbon. So these are the certain tests that is carried out to know the concentration of the organic matter present in it. So we are estimating what is the organic content present in the wastewater. And for that one, we have the BOD test. We can carry out the chemical oxygen demand, COD, or we can check the TOC, total organic carbon. So these are the certain tests is carried out. Now what is meant by biochemical oxygen demand? So here we are interested to know how much oxygen present in the wastewater after the treatment of it. Now after the treatment of it means, so it is a 5-day or 3-day test. Okay. So I will tell you the meaning of what is the 5-day test and what is the 3-day test and what is the temperature and other conditions are required. But here what we say, we are interested in first finding what is the total the oxygen present in it. So dissolved oxygen before the treatment means if we have the sample of wastewater and that sample we are diluted, I will tell you the latter one what is the process of it. But we are test what is the DO. Now what do you mean by DO? DO stands for the dissolved oxygen. How much dissolved oxygen present in a sample. Then we keep this sample in the BOD bottle. We have a certain process for it. I will tell you later afterward after this one. Okay. And in this case, we will, okay, can you hear me?
Yes, sir.
Okay, okay,
Yes, sir.
fine. Only I think that portion presentation दिसत नाही बरोबर.
नाही दिसत आहे. हो
Yes,
सर.
sir.
बरं 2 मिनिटं परत लावतो त्याला. Yes. Now it is visible.
Yes, sir.
Yes, sir.
Okay, fine. So what we do, we are initially note down what is the dissolved oxygen present before treatment, and then we will find out what is the dissolved oxygen after a treatment. After and before a treatment is what we have a BOD bottle. In that BOD bottle, we kept the samples, and obviously there is a bacteria present, and we know the what is the role of bacteria or the microorganisms. So they want a food for life, and for that one, what they do, they are decomposing the organic matter which is present in the wastewater. So they are starting the consumption of the organic matter, and they are converted that organic matter into the simple form. Simple form is obviously it is the carbon dioxide and the water, and the remaining some cells are generating. I will tell obviously in details afterward. So what is that BOD? It is the biochemical oxygen demand. Here we are trying to find initial oxygen and the final oxygen. Now what happened in that one? The organic, once the decomposition of the organic matter taking place, bacteria is not only consuming the organic matter, but also they are consuming the oxygen. Now where from they get the oxygen? Oxygen they get from the that wastewater, from that water. Okay, so obviously in that BOD bottle we are kept that bottle airtight, so that there is no additional oxygen from the atmosphere when goes into the bottle. So we pack it. Okay, whatever be the oxygen is present in that BOD bottle, only that can be consumed. Okay, now what happened here? Initially maybe have the 8 to 9 BOD the oxygen present before putting the sample into the BOD bottle, and then after say 5 days, it is happened that obviously the oxygen which is present in it, it is say 9 milligram per liter, and now there is a depletion takes place of the oxygen. Why there is a depletion? Because these bacteria are consuming the oxygen. Okay, and that we are related with the organic matter present. It means how much oxygen consumed is equals to the that much the organic matter present, because the organic matter is decomposed by the bacteria. Also bacteria is taking the oxygen, and we are correlate the organic matter and the oxygen, and that will give you a test which is called a biochemical oxygen demand. Means oxygen required to decomposing the organic matter. Now oxygen is what does the oxygen is helping to decomposing the organic matter? No, it is consumed by the microorganisms, bacteria which are present. But when they are consuming the oxygen, at the same time they are decomposing the organic matter. So there is a correlation between the oxygen and between the organic matter. So I am just telling you in a simple format of it. Okay, now let's see in details what exactly happening in that one. What is the aerobic treatment and aerobic one? So obviously we have taking this BOD test in the presence of the or the oxygen. Okay, that's why we call it is a biochemical oxygen demand. Same thing is for the chemical oxygen demand. Now in this case, what is the difference between the BOD and COD? Only the one difference. The one difference is what? Here we are using the bacteria and microorganisms for decomposition of the organic matter and how much oxygen is consumed. Now in the chemical oxygen demand, what we do? We are putting certain chemicals, okay, which can consume the oxygen. How they can consume is not we call the consumption, but they are making they give the reaction taking place, and whatever be the oxygen which is present in it and which is important for decomposition of the this organic matter can be utilized by the reaction, and on that basis we find out what will be the oxygen required, and on that basis we will find what is the organic matter present. Okay, so there is a correlation between consumption of the oxygen, declination of the oxygen is equals to the organic matter present in it. I am not talking about the inorganic matter. So these tests is not is used for the inorganic matter present. They are used only that these tests are carried out to know what is the organic matter content present in the wastewater. Is that clear? Now, let's see in detail about the biochemical oxygen demand, because whenever you go to the ETP plant, they have a certain data, the components are mentioned. The components, it may, they test the BOD, they test the COD, they test the pH, they test the TDS. TDS is what? It is total dissolved solid. They test the solid matter present. They test the what is the alkalinity. They test the what is the acidic, whether it is acidic or basic on the basis of the pH. They test the various inorganic matter present in it. Okay, and they have the chart. If you visited to the Phenolics industry and the TTP plant, so once you enter into the plant, before to that person, they will introduce about what are the components they are testing in the lab and what are the result from it. So day to day or time to time, they are mentioning the latest figure of it. Okay, fine. Now let's look the biochemical oxygen demand. Now what do you mean by biochemical oxygen demand? Already I said. So it is what? BOD can be defined as the oxygen required for a biochemical oxidation of the organic matter. So I told you in a simple language. Now here it is, a technical one is what? It is the oxygen required for a biochemical oxidation of the organic matter present in the water under the aerobic condition. Under the aerobic condition. So that is a BOD. So the oxygen required for a biochemical oxidation of the organic matter present in the water under the aerobic condition is called as the biochemical oxygen demand. Okay. So this test is based on the premises of all the biodegradable organic matter. As I said, it is the biodegradable organic matter. So the obviously again all the bacteria is not consuming the all type of the organic matter. Now if you have the microorganism or the bacteria and there is a oil content present in it. Now this bacteria doesn't decompose the oil or fat because they are consuming something different organic matter. So just like in our case, many of you may not be like some vegetables. Okay, why? Because on the basis of our taste, some of you are eat veg, some of you are eat non-veg, some are don't like non-veg. If there is if you have the plate of non-veg and you are eating only the veg, then definitely you are not consuming that one because that is not your food. Your food is what? It is a vegetables. Okay. Similarly, if certain animal or certain the person who is only eat the non-veg, if you put it the veg in his plate, then what happen? He will not eat that one because their food is different. Same thing is happen with the organic one. We can't say that which type of the food they like and which type of the food they don't like. But most of the times, the biodegradable food they are consumed. So here in the BOD test, though there is a certain toxic material is which is present, which we call the organic one, but they are not consuming that. They only consuming the biodegradable. Okay, so this is what we call, and obviously when they are oxidizing, what is the format? It is contain the CO2 and water. So if you see the reaction, if you we have the glucose, okay, so microorganisms are used here for the reaction and the oxygen is required. Okay, so when you are taking this glucose plus oxygen, microorganisms are here and there is a formation of the carbon dioxide and the water. What is the end product after the decomposition? It is the carbon dioxide and water. But obviously here is again the plus point. Definitely we will see in later in the next reaction what exactly other cells are formed or are the microorganisms formed and how the reaction is carried out. But here we can, with the help of this reaction, we can easily understood how much theoretical oxygen demand. Now we know that very well. We know very well the reaction balance. Okay, there is one molecule of the glucose and six molecules of the oxygen. It will give the six molecules of the CO2 and the six molecules of the water. Okay, now here how much oxygen demand? Now, if you want to convert an 100% conversion of this glucose into the carbon dioxide and into the water, then how much oxygen is consumed? Even you done that the theoretical calculation of this in the process calculation subject. Okay, when you are studying in the second year. So here the theoretical oxygen demand we can calculate on the basis of what? Oxygen demand is equals to the gram of oxygen used, this one, upon the gram of carbon oxidized. How much gram of oxygen is used and how much gram of carbon dioxide, oxidized, means this carbon oxidized. So here if you see the standard reaction, then it will have a 192 by 72. It will give the 2.67 gram per gram of the carbon. So how much oxygen required? It is to consume the 1 gram of carbon, I require the 2.67 gram of the oxygen. Okay, for the consumption of the single carbon, I need a 2.67 gram of the oxygen. That should be the present in it. Then only there is a complete reaction takes place and formation of the carbon dioxide and the water, which is a simple or the end product. Okay, so here we can say that the actual BOD will be less than the theoretical oxygen demand. Now here is the one statement that what actual BOD will be less than the theoretical oxygen demand due to the incorporation of the sum of the carbon into the newly synthesized bacterial cell. As I said, so this is what we call the theoretical one. Okay, so this much is the glucose, this much is the oxygen, it will give this product. But when we are dealing with the microorganisms, at the same time, the microorganism growth takes place. Means if you have the 10 microorganisms initially present and they are consuming the food, but at the same time they are growing. Okay, so they produce they reproduce the another the microorganisms. So from the 10 it may have the 15. This 15 may be again formation of the 20, this may be 20 again formation of the 30. So there is a continuous growing of the microorganisms. When there is a continuous growing of the microorganisms, in that case what happened? The new cells are formed. New cell is what? A new microorganism is produced. Okay, so this, because of that one, what happened? A new cells are formed. Some microorganisms get dead. Some new microorganisms are formed. So this is the cycle continues going on till there is a present of the organic matter present. It means initially the growing of the microorganism is very high. So within a 2 days from the 10 it may have up to the 100, and then slowly the glucose or the organic matter which is present are vanishing because there is a limited, say 1 gram of the organic matter present, and there 10 number of microorganisms are present and they are consuming the food, and they are also reproduce the microorganisms. And what happened? Initially the growing rate is very high, the decomposition rate is very high, but after the time the food is vanished, it is not available as that quantity, and whatever be the microorganisms which are grow nowadays are becoming dead because of the unavailability of food, because of the life cycle is over. Okay, so because of that what happened? A new cells is also formed and it will be the decay, and for that's why the actual BOD is always less than the theoretical BOD. So that is the reason why there is a if the question asks why there is a theoretical oxygen demand is less than the actual BOD, or what is the why the actual BOD will be sorry, why the actual BOD will be less than the theoretical one. The reason behind is this. Okay, so here what we do, the water sample incollated. Okay, so already I give the explanation about it, how the things are happen, how the organisms utilize the oxygen, and what will happen. Okay, the process consuming the organic matter and all. So that in this test, obviously we will focusing on the test now. Now, what is the reason that we are why we are performing the BOD test? What is the reason behind it? One of the reason is what? To determine the quantity of oxygen required for a biochemical stabilization of the organic matter. Why we are conducting the test? We are conducting the test just to know what is the oxygen required for a biochemical stabilization of organic matter. It means stabilization means what? End product converted of organic matter into a CO2 and water. That is the one case. Second is what? Determine the suitability of the biological treatment method. As I said earlier, to determine the what is the suitability of the treating the wastewater, what type of the process that I have to choose for giving the treatment to this. And for that one, we need to know what is the COD to BOD ratio. So here it is mentioned. What is the COD to BOD ratio? This is very important. Okay. So it is this one. It is what? COD to BOD ratio. On that basis, we will choose what type of the process or the treatment methods that we need to give. So let's see what is the ratio. If the ratio is given about less than 2, we are testing the sample. BOD, what is the value of BOD? Same sample we test what is the value of COD. And then when we got the value of COD and we got the value of BOD, we take the ratio of it. And if that ratio is less than 2, then this is the suitable for giving a biological treatment. Means we can use the activated sludge process, we can use the oxidation pond, we can use the packed bed reactor, aerobic one, we can use the trickling filter. So different type of the treatments that we are giving, which having the biological treatment. Means the bacteria and microorganisms are used for that process. Okay, fine. And now, if the ratio is more than 2 and less than 6, if you see here, if the ratio is more than 2, okay, and less than 6, in this case what we do, we can give the biological treatment, but one thing we have to do is that we need to dilute that particles, we need to dilute the solution, the wastewater, that is one thing, or we need to add certain glucose or certain organic matter additionally, so that there is a bacterial growth takes place. Because some of the biodegradable is very hard for that microorganisms to consume. And for that one, what we do, we are giving the additional dose. Just like I would like to give the example, when we are ill, okay, that time what we do, we are taking the extra dose of the glucose. Because of the weakness, because of the illness, what happens, there is a weakness, and our energy is not charging, or whatever we food we eat may not give that much energy, or they may not generate that much energy. So what we do, we are taking the extra dose of the glucose. So we use the glucose, we are taking with the food. Food is the one section and the glucose is another. Because of that, what happened, we are charged. Okay, we will charge because of the addition of the glucose in our stomach, in our body. So same thing is happened if the BOD contain a COD ratio, COD to BOD ratio, if it is more than 2 to 6. In that cases, we are adding certain things, certain foods. Additionally, the things which is the bacteria and microorganisms are like. Okay, many times when we don't have a good food, or when we don't have the vegetable, we don't like the vegetables. That time what we do, we additionally we are taking some another food, another vegetables, so that it can be match up with that. Okay, many times even we are demanding to our mother that we don't want this one, we don't like, so you give me something else. So mother give you some small which you like, and obviously the vegetable which she made, so you have to eat that one with the combination of it. Same thing is happened with this. Okay, and if the COD to BOD ratio is greater than the 6, if it is greater than the 6, in that cases we are skip the biological treatment, and here we need to use the another suitable treatment for it. So I can use some chemical treatments or some additionally technology that I have to use, the process I have to use, and treating that one. Okay. So here we are looking what we are giving the reason why we are doing a test or why we are conducting the test. Uh, Third part is the monitor efficiency of the process. To monitor the efficiency of the process, it means when you are establishing a plant, okay, you are sending the wastewater into the plant, and there is a discharge of the plant, the waste from the wastewater from the plant, and if you don't know how much is the in and how much is the out, you can't monitor it. So initially you have to take what is the influent to the wastewater treatment. Just let's take the example of the activated sludge process. Okay, we have the plant of activated sludge process, and we are giving the influent means in. So in that case we note down what is the BOD content when it is enter into the activated process, and what will be the value of the BOD when it is out from the process. And by telling this one, we can say that yes, there is a treatment is going on properly, and it is as per our norms and as per the efficiency of the plant. Means if the BOD having the 600, that is enter, and we know that we want up to the 40, that is the capacity of the plant, that up to the 40 they can reduce. So you check it whether the value of the effluent which is come from the activity sludge process, is it a 40? If it is a 40, then yes, we say that efficiently the plant is working. Okay, now I can easily discharge the water into the river side. So that is what a monitoring efficiency of the process. And the last one is to determine the compliance with the wastewater discharge permits. So obviously we have to know the norms of the government, and we have to follow the norms of the pollution control boards. And again we have to look out whether as per the pollution control board, as per the WHO standards, as per the BIS standard, is it the plant is treating that one or not. Okay, so that's why we are conducting this the BOD test. Now let's say what happened here in detail about the reaction, what reaction is carried out. Okay, you learn the chemical reaction engineering, and you know very well how the reactions carried out. Even in the chemistry you learn or the things. So what is the reaction? The reaction is organic matter, oxygen, and the catalyst we call it is the microorganisms, and the end product already we see in the previous reaction, it is CO2 and water. And as I said, why there is the actual BOD is less than the theoretical one, the answer being is what? The new cells are formed, means the growing of microorganisms, and there is a stable product. Stable product is what? Some organic matter which we are consuming have some stability. Okay, so all these things are generated when the reaction is carried out. Okay, and apart from this there may have the sulfate, orthophosphate, and nitrates are formed. That is the end product that we call the stable end product, as it may have the nitrate, it may have the sulfate, it may have the CO2. Okay, so this, that's why we say that the actual BOD is less than the theoretical one. Now this is the reaction which is carried out, it is in presence of oxygen. Now, up to the oxygen present in the wastewater in the plant treatment, then this reaction is carried out. But if there is a deficiency of the oxygen, then the reverse cases happen. Rather than the aerobic, there is an anaerobic treatment takes place, anaerobic for the reaction taking place, and the product may be the different. And what will be the product for that one? If there is a insufficient, let's say the reaction contains still the sufficient DO. So this reaction is carried out up to the DO, but after that one the reverse is happened, and there is a fermentation takes place, and that can be takes place with the in the anaerobic process. So here the microorganisms, organic matter, these microorganisms, which microorganisms? Which are live in absence of what we call in absence of oxygen. They can live in absence of the oxygen. That microorganisms are decomposing. Just like a fermentation. In the fermentation, what happens? They don't require the oxygen. They are the bacteria which can consume they require the oxygen, but they can take place with the help of the reaction. So we call anaerobic treatment. So here, organic matter, microorganisms, formation of the CO2, the CO2 is there definitely, and the one product which is formed, it is the methane. Also, they form the new cells. Also, they have some stable the product which is called ammonia and the hydrogen sulfide. But this reaction is carried out when there is a insufficient or the total oxygen it get consumed by the microorganisms, and if the organic matter still present in that one and we are not introducing the oxygen, in that cases this reaction is carried out. Okay, now quickly we will look about the BOD test, how the BOD test is carried out. Okay, so as it's a very slow process, it may require the 5 days, it may require the 3 days, or more than the 5 days too. But maximum we are taking a 5 day test for a 20 degrees Celsius one. We have the incubator. We use the BOD bottle. In that BOD bottle, diluted wastewater we are putting into that sample in the BOD bottle. We close that one, pack it, so that there is no the additional air from the atmosphere to enter into the BOD bottle. We are putting the culture, that is some bacteria's culture. We put it some glucose, some the food contents for the bacteria at the initial stage. Okay, so that there is the growing of the microorganism takes place, and we will test what will be the DO present initially when before we don't have without treatment, means when the BOD bottle initially when we put it the sample and we look it, and after a 5 days when the bottle get closed, it is airtight, and after 5 days when we are collecting that bottle and again we collect the sample from the BOD bottle and we test it and we found what will be the DO present in it. So here what happened at the initial stage, definitely the rate of reaction is very high, and after that one it will be the decline. Okay, I think the time is almost over. This part that we will see in tomorrow's lecture, because this is very important one. Once we understood what is meant by BOD, okay, what is the COD, then it's very easy for you to just understood the concept about the wastewater treatment. Okay, is there anybody having any doubt?
No, sir.
Okay, fine. So shall we stop here?